students, welcome to the PD Life Science Academy for the Happy Learning. NEET एक्झामचे प्रिपरेशन जे विद्यार्थी करत आहेत त्यांच्यासाठी बेसिक कन्सेप्ट लर्निंग आणि लिंकिंग ऑफ कन्सेप्ट हे जेवढे गरजेचे आहे तेवढेच NCERT बुकमधील चॅप्टरचे कंटेंट रीड करणे आवश्यक आहे. हे करत असताना फक्त एक रीडिंग करून चालणार नाही, मल्टिपल टाइम्स रिव्हिजन होणे गरजेचे आहे. यात टाळाटाळ करणे चांगले नव्हे. आपण सेट केलेले टार्गेट अचीव करण्यासाठी PD लाईफ सायन्स अकॅडमीने सर्व स्टुडंटसाठी जे नीट एक्झामचे प्रिपरेशन करीत आहेत त्यांच्यासाठी 21 डे रीडिंग चॅलेंजची सुरुवात केली आहे. रोज एका चॅप्टरचे रीडिंग आपण कंप्लीट करणार आहोत. हे चॅलेंज ज्या विद्यार्थ्यांनी ॲक्सेप्ट केले आहे त्यांनी त्यांचे डेली टार्गेट अचीव केल्यावर कॉमेंट करून सांगायचे आहे. सो लेट्स गेट रेडी फॉर टेकिंग रीडिंग चॅलेंज. ऑल द व्हेरी बेस्ट. थँक यू. 21 डे बायोलॉजी रीडिंग चॅलेंज डे 11 रीडिंग चॅलेंज चॅप्टर नंबर 11 फ्रॉम NCERT बायोलॉजी टेक्स्टबुक ऑफ 11th standard. Name of chapter is a transport in plants. Have you ever wondered how the water reach the top of the tall trees, or for that matter how and why substances move from one cell to the other? Whether all substances move in a similar way in the same direction, and whether the metabolic energy is required for the moving substances. Plants need to move the molecules over very long distances, much more than the animal do. They also do not have the circulatory system in place. Water taken up by the root has to reach all parts of the plant, up to the very tip of the growing stems. The photosynthesis or a food synthesized by the leaves have also been moved to the all parts, including the root tip embedded deep inside the soil. Movement across the short distances, say within the cell, across the membrane, and from the cell to cell within the tissue has also to take place. To understand some of the transport processes that take place in the plant, one would have to recollect once the basic knowledge about the structure of the cell and the anatomy of the plant body. We also need to revisit our understanding of the diffusion besides gaining some knowledge about the chemical potential and the ions. When we talk of the movement of the substances, we need first to define what kind of the movement we are talking about, and also what substances we are looking at. In flowering plant, the substances that would need to be transported are the water, minerals, nutrients, mineral nutrients, organ nutrients, and the plant growth regulators. Over small distance, substances move by the diffusion and by the cytoplasmic streaming, supplemented by active transport. Transport over a long distance proceeds through the vascular system, that is the xylem and the phloem, and is called as a translocation. An important aspect that needs to consider the direction of the transport. In rooted plant, the transport in the xylem, that is of water and the minerals, is essentially unidirectional from roots to the stem. Organic and the mineral nutrition, however, undergoes the multidirectional transport. The organic compounds synthesized in the photosynthetic leaves are exported to all parts of the plant, including the storage organ. From the storage organ, they are later re-exported. The mineral nutrients are taken up by the roots and transported upward into the stem, leaves, growing region. When any plant part undergoes the senescence, the nutrients may withdraw from such regions and move in the growing parts. Hormones or the plant growth regulators and the other chemical signals are also transported through a very small amount and sometimes in the strictly polarized or the unidirectional manner from where they are synthesized to the other part. Hence, in flowering plant there is a complex traffic of the compounds, but probably a very orderly moving in the different directions, each organ receiving some substances and giving out some others. Now the 11.1, that is means of the transport. 11.1.1 is the diffusion. The movement by diffusion is a passive and may be from the one part of the cell to the other and from the cell to cell or over a short distance, say from the intercellular spaces of the leaf to the outside. No energy expenditure takes place. In diffusion, the molecule moves in the random fashion and the net result being the substances moving from the region of the higher concentration to the region of the lower concentration. Diffusion is a very slow process and is not dependent on the living system. Diffusion is obviously a gaseous and the liquid, but diffusion in a solid is more likely rather than the solids. The diffusion is a very important in the plant since it is only means for the gaseous movement within the plant body. Diffusion rate are affected by the gravity of the concentration, the permeability of the membrane separating them, and the temperature and the pressure. Now the 11.1.2 is a facilitated diffusion. As pointed out earlier, a gradient must already be present for the diffusion to occur. The diffusion rate depends on the size of the substances. Obviously, the smaller substances diffuse faster. The diffusion of any substances across a membrane also depends on its solubility in the lipid, the major constituents of the membrane. The substances soluble in the lipid diffuse through the membrane faster. The substances that have a hydrophilic moiety find it difficult to pass through the membrane. Their movement has to be facilitated. The membrane protein provides the site at which such molecule cross the membrane, and they do not set up the concentration gradient. A concentration gradient must already be present for the molecule to diffuse even if facilitated by the proteins. This process is called as a facilitated diffusion. In facilitated diffusion, the special protein helps move substances across the membrane without expenditure of the ATP energy. Facilitated diffusion cannot cause the net transport of the molecule from the low to the high concentration. This would require the input of the energy. Transport rate reaches a maximum when all protein transporters are being used, that is the saturation. Now the facilitated diffusion is a very specific. It allows the cell to select the substances for the uptake, and it is sensitive to the inhibitors which reacts with the protein side chain. The proteins form a channels in the membrane for the molecules to pass through. Some channels are always open, others can be controlled. Some are the large, allowing a variety of the molecules to cross. The porins are the proteins that forms the large pores in the outer membrane of the plastids, mitochondria, and some bacteria, allowing the molecule up to the size of the small proteins to pass through. Now figure shows the extracellular molecule bound to the transport protein. The transport protein then rotate and release the molecule inside the cell. For example, the water channel made up of the 8 different types of the aquaporins. Now the 11.1.2.1 is a passive symport and the antiport. Some carrier or the transport protein allows the diffusion only if 2 types of the molecules move together. In symport, both molecules cross the membrane in the same direction. In the antiport, they move in the opposite direction. When a molecule moves across the membrane, independent of other molecules, the process is called as a uniport. Now the 11.1.3 is active transport. Active transport uses the energy to transport and pump the molecule against the concentration gradient. Active transport is carried out by the specific membrane protein. Hence, the different proteins in the membrane plays a major role in both active as well as passive transport. Pump are the proteins that use the energy to carry the substances across the cell membrane. These pumps can transport the substances from low concentration to the high concentration, that is uphill the transport. The transport rate reaches the maximum when all protein transporters are being used or are saturated. Like enzymes, the carrier protein is a very specific in what it carries across the membrane. These proteins are the sensitive to the inhibitor and reacts with the protein side chain. Now the 11.1.4 is a comparison of the different transport processes. Table gives the comparison of the different transport mechanism. The protein in the membrane are the responsible for the facilitated diffusion and the active transport, and hence shows the common characteristic of being highly selective. They are liable to saturate, respond to inhibitors, and are under the hormonal regulation. But diffusion, whether facilitated or not, take place only along a gradient and do not use the energy. Now the properties, in simple diffusion require a specific membrane protein. There is no any requirement in simple diffusion. There is no any requirement of the special membrane protein, then highly selectivity, then the transport saturation, uphill transport, and the ATP energy. But in case of facilitated transport, there will be required a special membrane protein. It may be highly selective, and the transport is a saturated form. But there is no need of a uphill transport and the requirement of the ATP, that is the energy molecule. But for the active transport, all factors or all properties must be there, that is there is a requirement of the special membrane protein. They are highly selective. The transport is a saturates, then the uphill the transport, and it requires the ATP in the form of energy. Now the 11.12 is a plant water relation. Water is essential for all physiological activities of the plant and plays a very important role in all living organisms. It provides a medium in which most substances are dissolved. The protoplasm of the cell is nothing but the water in which the different molecules are dissolved and the several particles get suspended. The watermelon has over the 92% water, and the most herbaceous plants have only about the 10 to the 15% of its fresh weight as a dry matter. Of course, the distribution of the water within a plant varies. Woody part have relatively very little water, while the soft part mostly contains a water. A seed may appear dry, but it still has a water, otherwise it would not be alive and respiring. Terrestrial plants take up the huge amount of the water daily, but most of it lost to the air and through the evaporation from the leaves, that is the transpiration. The mature corn plant absorbs almost 3 liters of the water in a day, while the mustard plant absorbs the water equals to its own weight in about the 5 hours. Because of this high demand of the water, it is not surprising that water is often a limiting factor for the plant growth and productivity in both agriculture and the natural environment. Now the 11.2.1 is the water potential. To comprehend plant water relations, to understanding of the certain standard terms is necessary. That is the water potential, that is the psi W, is a concept fundamental to understanding the water movement. Now the solid potential, that is the psi S, and the pressure potential, that is the psi P, are the two main components that determine the water potential. Water molecule possess a kinetic energy. In liquid and the gaseous form, they are in the random motion, that is both rapid and the constant. The greater the concentration of the water in the system, the greater is its kinetic energy or the water potential. Hence it is obvious that a pure water will have a greatest water potential. If two systems containing the water are in contact, random movement of the water molecule will result in the net movement of the water molecule from the system with a higher energy to the one with the low energy. Thus, the water will move from the system of a system containing the water at a higher water potential to the one that having the low water potential. This process of the movement of the substances down the gradient of the free energy is called as a diffusion. The water potential is denoted by the Greek symbol that is the psi and is expressed in the pressure unit such as the Pascal. By the convention, the water potential of the pure water at a standard temperature which is not under any pressure is taken to be 0. If some solutes is dissolved in the pure water, the solution has a fewer free water molecule and the concentration, that is the free energy of the water, decreases, reducing its water potential. Hence all solutes have the lower water potential than the pure water. The magnitude of this lowering due to the dissolving of the solute is called as a solute potential, that is the psi s. Psi s is always negative. The more solute molecules, the lower or the more negative is the psi s. For a solution at atmospheric pressure, that is the water potential, that is the psi w is equal to solute potential, that is the psi s. If the pressure greater than the atmospheric pressure is applied to the pure water or a solution, its water potential increases. It is equivalent to the pumping water from one place to the another. You can think of any system in our body where the pressure is a build-up. The pressure can be built up in a plant system when the water enters a plant cell due to the diffusion causing a pressure build up against the cell wall. It makes the cell turgid. This increase in the pressure potential. The pressure potential is usually positive, through in the plant negative potential or a tension in the water column in the xylem plays a major role in the water transport up a stem. The pressure potential is denoted by the psi p. The water potential of the cell is affected by both solute and the pressure potential. The relationship between them is as follows, that is the psi w is equal to psi s plus psi p. Now the 11.2.2, that is the osmosis. Now the plant cell is surrounded by the cell membrane and the cell wall. The cell wall is freely permeable to the water and the substances in the solution, hence it is known as a barrier. It is not barrier to the movement. In plants, the cells usually contains the large central vacuoles and whose content the vacuolar sap or contributes to the solute potential of the cell. In a plant cell, the cell membrane and the membrane of the vacuole, the tonoplast, together are the important determinants of the movement of the molecules in or out of the cell. Now the osmosis is the term used to refer specifically to diffusion of the water across the differentially or selectively permeable membranes. Osmosis occurs spontaneously in response to the driving forces. The net direction and the rate of the osmosis depends on both the pressure gradient and the concentration gradient. The water will move from its region of the higher chemical potential or the concentration. It is the region of the lower chemical potential until the equilibrium is reached. At equilibrium, the two chambers should have nearly the same water potential. You may have made a potato osmometer in your earlier classes in the school. If the potato tuber is placed in the water enters the cavity in the potato tuber containing a concentrated solution of the sugar due to osmosis. Now study in the figure in which the two chambers, that is the A and the B, containing a solution are separated by the semi-permeable membranes. Now first is the solution of which chamber. There are some questions arises. Let us discuss another experiment where the solution of the sucrose in the water taken in the funnel is separated from the pure water in a beaker by the selectively permeable membrane. You can get this kind of the membrane in the egg. Remove the yolk and albumin through the small hole at the one end of the egg and place the shell in a dilute solution of the hydrochloric acid for a few hours. The egg shall get dissolved, leaving the membrane intact. The water will move into the funnel, resulting in a rise in the level of the solution in the funnel. This will continue till the equilibrium is reached. In case the sucrose does not diffuse out through the membrane, will this equilibrium is ever reached, the external pressure can be applied from the upper part of the funnel such that no water diffusion into the funnel through the membrane. This pressure requires to prevent the water from the diffusing is in fact the osmotic pressure, and this is the function of the solute concentration. More the solute concentration, greater will be the pressure required to prevent the water from diffusing in. Numerically, the osmotic pressure is equivalent to the osmotic potential, but the sign is opposite. The osmotic pressure is a positive pressure applied, while the osmotic potential is a negative. Now the 11.2.3 is the plasmolysis. The behavior of the plant cell or the tissues with regard to the water movement depends on the surrounding solution. If the external solution balance the osmotic pressure of the cytoplasm, it is said to be the isotonic. The external solution is more diluted than the cytoplasm, it is known as a hypotonic, and if the external solution is a more concentrated, it is called as a hypertonic solution. Cell gets swell in a hypotonic solution and shrink in the hypotonic ones. Now the plasmolysis occurs when the water moves out of the cell and the cell membrane of the plant cell gets shrink away from its cell wall. This occurs when the cell or the tissues is placed in the solution that is the hypertonic and has a more solute to the protoplasm. Water moves out. It is the first loss from the cytoplasm and then from the vacuole. The water when draws out of the cell through the diffusion into the extracellular, that is the outside the cell, the fluid causes the protoplasm to shrink away from the wall. The cell is said to be the plasmolized. The movement of the water occur across the membrane, moving from an area of the high water potential, that is the cell, to the area of the low water potential, that is outside the cell. What occupies the space between the cell wall and the shrunken protoplast in the plasmolized cell? When a cell or a tissue is placed in the isotonic solution, there is no net flow of the water towards the inside or outside. If the external solution balance the osmotic pressure of the cytoplasm is said to be the isotonic. When the water moves into the cell and out of the cell and are equilibrium, the cell is said to be the flaccid. The process of the plasmolysis is usually reversible when the cell are placed in the hypotonic solution, that is the high water potential or a dilute solution, as compared to the cytoplasm. Water diffuses into the cell, causing the cytoplasm to build up a pressure against the wall. That is called as a turgor pressure. The pressure exerted by the protoplast due to the entry of the water against the rigid wall is called as a pressure potential, that is the psi p. Because of the rigidity of the cell wall, the cell does not rupture. This turgor pressure is ultimately responsible for the enlargement and the extension growth of the cell. Now the 11.2.4 is the imbibition. Imbibition is a special type of the diffusion when the water is absorbed by the solid or colloids, causing them to increase in the volume. The classical example of the imbibition are the absorption of the water by the seeds and the dry woods. The pressure that is produced by the swelling of the wood has been usually by the prehistoric man to split the rocks and the boulders. If it were not for the pressure due to imbibition, the seedling would not have been able to emerge out of the soil into the open. They probably would not have been able to establish. Imbibition is also a diffusion, since the water movement is along the concentration gradient. The seeds and the other such material have also almost no water, hence they absorb the water easily. The water potential gradient between the absorbent and the liquid imbibe is essential for the imbibition. In addition, for any substances to imbibe any liquid, affinity between the absorbent and the liquid is also a prerequisite. Now the 11.3 is the long distance transport of the water. At some earlier stages, you might have carried out an experiment where you had placed a twig being a white flower in a color water and had a watch it turns a color. On examining the cut end of the twig after a few hours, you have note the region through which the color water is moved. That experiment very easily demonstrated that the path of the water movement through the vascular bundle, more specifically the xylem. Now we have to go further and try to understand the mechanism of the movement of the water and the other substances of a plant. Long distance transport of the substances within the plant cannot be by diffusion alone. Diffusion is a slow process. It can account for a only short distance movement of the molecule. For example, the movement of the molecule across the typical plant cell, that is about the 50 micrometer, takes approximately the 2.5 seconds. At this rate, can you calculate how many years it would take for the movement of the molecule over a distance of the 1 meter within a plant by diffusion alone? In a large and a complex organism, often substances have to be moved to long distances. Sometimes the site of the production or the absorption and the site of the storage are too far from each other. Diffusion or the active transport would not suffice. The special long distance transport system becomes necessary so as to move substances across the long distance and at much faster rate. The water and the mineral and the food are generally moved by the mass or a bulk flow system. The mass flow is the movement of the substances in a bulk or in a mass from the one point to the another as a result of the pressure differences between the 2 points. It is a characteristic of the mass flow that substances, whether in a solution or in a suspension, are swept along at the same pace as in the flowing river. This is unlike diffusion, where the different substances move independently depending on their concentration gradient. Bulk flow can be achieved either through the positive hydrostatic pressure gradient, for example the garden hose, or negative hydrostatic pressure gradient, that is for example the suction through the straw. Now the bulk movement of the substances through a conducting or a vascular tissues of the plant is called as a translocation. Do you remember the studying a cross section of the root, stem and leaves of the higher plants and studying the vascular system? The higher plants have highly specialized vascular tissues, the xylem and the phloem. Xylem is associated with the translocation of the mainly the water, mineral salt, some organic nitrogen and the hormones. From roots of the aerial parts of the plant, the phloem translocates a variety of the organic and the inorganic solids, mainly from the leaves to the other parts of the plant. How do the plant absorb the water? We know that the root absorbs most of the water that goes into the plant, and obviously that is why we apply the water to the soil. and not on the leaves. The responsibility of the absorption of the water and the mineral is more specifically the function of the root hairs that are present in the millions at the tip of the root. The root hairs are the thin wall slender extensions of the root epidermal cells that greatly increase the surface area of the absorption. Now the water is absorbed along with the mineral solute by the root hairs purely by the diffusion. Once the water is absorbed by the root hair, it can move deeper into the root layer by two distinct pathways, that is, one is the apoplast pathway and second is the symplast pathway. The apoplast is a system of the adjacent cell wall that is continuous throughout the plant except at the Casparian strip of the endodermis in the root. The apoplastic movement of the water occurs exclusively through the intercellular spaces and wall of the cell, that is, the movement through the apoplast does not involve a crossing the cell membrane. The movement is depend on the gradient. The apoplast does not provide any barrier to the water movement, and the water movement is through the mass flow. As the water evaporates into the intracellular spaces or the atmosphere, the tension develops in the continuous stream of the water in the apoplast. Hence, the mass flow of the water occurs due to the adhesive and the cohesive properties of water. The symplastic system is a system of the interconnected protoplast. Neighboring cells are connected through the cytoplasmic strands that extend through the plasmodesmata. During the symplastic movement, the water travels through the cell and their cytoplasm. Intercellular movements is through the plasmodesmata. Water has to enter the cell through the cell membrane, hence the movement is relatively slower. The movement is again down a potential gradient. Symplastic movement may be aided by a cytoplasmic streaming. You may have observed the cytoplasmic streaming in cell of the hydrilla leaf. The movement of the chloroplast due to the streaming is easily visible. Most of the water flow in the root occurs via a apoplast, since the cortical cells are loosely packed, hence offer no resistance to the water movement. However, the inner boundaries of the cortex, the endodermis, is impervious to the water because of the bands of the suberized matrix that is called as the Casparian strips. The water molecules are unable to penetrate the layer, so they are directly to the wall region that are not suberized into the cell proper through the membrane. The water then moves through the symplast and again crosses a membrane to reach the cell of the xylem. The movement of the water through the root layer is ultimately symplastic in the endodermis, and this is only way water and the other solute can enter the vascular cylinder. Once inside the xylem, water is again free to move between the cell as well as through them. In a young root, the water enters directly into the xylem vessels and or tracheids. There is no non-living conduits, and so as a part of the apoplast. The path of the water and the mineral ions into the root vascular system is summarized in figure. Some plants have the additional structures associated with them and helps in the water, that is water and the minerals absorption. A mycorrhiza is a symbiotic association of the fungus with the root system. A fungal association, that is or a fungal filaments form a network around the young root, or they penetrate the root cells. The hyphae have a large surface area that absorb the mineral, ions and water from the soil from much larger volume of the soil that perhaps a root cannot do. The fungus provide a minerals and the water to the root. In the turns of the root provide a sugar and the nitrogen containing compounds to the mycorrhiza. Some plants have an obligate association with the mycorrhiza. For example, the pinus seed cannot germinate and establish without a presence of the mycorrhiza. Now the 11.3.2 is a water movement up a plant. We look at how plant absorb the water from the soil and move it into a vascular tissue. We now have to try to understand how this water is transported to a various parts of plant. Is the water movement is active or is it still a passive? Since the water has to move up the system against the gravity, what provides the energy for this? Now the 11.3.2.1 is a root pressure. As various ions from the soil are actively transported into the vascular tissues of the roots, the water flows, that is its potential gradient, and increases the pressure inside the xylem. The positive pressure is called as a root pressure and can be responsible for pushing up a water to small heights in stem. How can we see that the root pressure exists? Choose a small soft stem plant, and on a day when there is a plenty of the atmospheric moisture, cut a stem horizontally near the base with a sharp blades early in the morning. You will see the drops of the solution ooze out of the cut. This comes out due to the positive root pressure. If you fix the rubber tube to the cut stem as the sleeves, you can actually collect and measure the rate of the exudation and also determine the composition of the exudates. Effect of the root pressure is also observable at night and early morning when the evaporation is low and excess water get collected in the form of the droplet around the special opening of the veins near the tip of the grasses, grass blades, and the leaves of many herbaceous parts. Such a water loss in its liquid phase is known as a guttation. Root pressure can be best only provide a modest push in an overall process of the water transport. They obviously do not play a major role in the water movement up a tall trees. The greatest contribution of the root pressure may be re-establish a continuous chain of the water molecule in the xylem, which often break under the enormous tension created by a transpiration. Root pressure does not account for the majority of the water transport. Most plants meet their need by transpiration pool. Now the 11.3.2.2 is a transpiration pool. Despite the absence of the heart or a circulatory system in a plant, the upward flow of the water through a xylem in a plant can achieve fairly high rates, up to the 15 meters per hour. How is this movement is accomplished? A long standing question is whether water is a push or pull through the plant. Most researchers agree that the water mainly pull through the plant, and that is the driving force for this process is a transpiration from the leaves. This is referred to as the cohesion tension transpiration pull model of the water transport. But what generates this transpiration pull? The water is a transient in the plant. Less than the 1% of the water is reaching the leaves is used in the photosynthesis and the plant growth. Most of it is lost through the stomata in the leaves. This water loss is known as a transpiration. You have studied a transpiration is in the earlier class by enclosing a healthy plant in a polythene bag and observing a droplets of the water form inside the bag. You could also study the water loss from the leaves using the cobalt chloride paper, which turns color on absorbing the water. Now the 11.4, that is the transpiration. Transpiration is the evaporating, the loss of the water by the plant. It occurs only through the stomata, that is in singular word is a stoma. Beside the loss of the water vapor is a transpiration, that is the exchange of the oxygen and the carbon dioxide in the leaves also occurs through these stomata. Normally the stomata are open in a daytime and closed during the night time. Immediate cause of the opening and the closing of the stomata is a change in the turgidity of the guard cell. The inner wall of each guard cell towards the pore or a stomatal aperture is a thick and the elastic. When the turgidity increase within the outer guard cell and flanking each stomatal aperture or a pore, a thin outer wall bulge out and force the inner wall into the crescent shape. Opening of the stoma is also added due to the orientation of the microfibril in the cell wall of the guard cell. The cellulose microfibrils are oriented radially rather than the longitudinally, making it easier for the stomata to open. When the guard cell get lose the turgidity due to the water loss or water stress, the elastic inner wall again their original shape and the guard cell becomes a placid and stomata get closed. Usually the lower surface of the dorsiventral, often the dicotyledonous leaf, has the greater number of the stomata, while in a isobilateral, that is often a monocotyledonous leaf, they are about equal on both surfaces. Transpiration is affected by the several external factors like the temperature, light, humidity, wind speed. The plant factors that affect the transpiration include the number and the distribution of the stomata, percent of the open stomata, then the water status of the plant and a canopy structure, etc. The transpiration driven ascent of the xylem sap depends on mainly on the following physical properties of the water. That is, first is a cohesion, the mutual attraction between the water molecule. Then second is adhesion. Adhesion means the attraction of the water molecule to the polar surface, such as the surface of the tertiary element. And third is the surface tension, that is the water molecule are attracted to each other in the liquid phase more than to water in the gas phase. These properties gives the water high tensile strength, that is, and the ability to resist the pulling force, and the high capillarity, that is the ability to rise in a thin tube. In plants, capillarity is added by the small diameter of the tertiary element, the tracheids and the vessel element. The process of the photosynthesis requires the water. The system of the vessel, xylem vessel from the root to the leaf veins can supply the needed water. But what force does a plant use to move the water molecule into the leaf parenchyma cells where they are needed? As the water evaporates through the stomata, since the thin film of the water over a cell is continuous, it results in a pulling of the water molecules by the molecule into the leaf from the xylem. Also, because of the lower concentration of the water vapor in the atmosphere as compared to the substomatal cavity and the intercellular spaces, the water diffuses into the surrounding air and this creates a pull. The measurement reveals that the force generated by the transpiration can create a pressure sufficient to lift a xylem size a column of the water over a 130 meters height. Now the 11.4.1 is a transpiration and the photosynthesis, a compromise. Transpiration has a more than one purpose. It creates a transpiration pool for the absorption and the transport of the plant, then it supplies the water for the photosynthesis, transport the minerals from the soil to all parts of the plant. It cools the surface, leaf surface, and sometimes the 10 to 15 degrees by evaporating the cooling. It maintains the shape and the structure of the plant by keeping the cell turgid. And actively photosynthesizing the plant has the insatiable needs for the water. Photosynthesis is limited by the availability of the water, which can swiftly depleted by the transpiration. The humidity of the rainforest is largely due to this vast cycling of the water from the root to the leaf to the atmosphere and back to the soil. The evolution of the C4 photosynthetic system is probably one of the strategies for maximizing the availability of the CO2 while minimizing the water loss. The C4 plants are twice as efficient as the C3 plants in terms of the fixing the carbon dioxide that is making in the sugar. However, the C4 plant loses only the half as much of the water as a C3 plant for the same amount of the CO2 fixation. Now the 11.5 is uptake and transport of the mineral nutrients. Now a plant obtain their carbon and the most of their oxygen from the CO2 in the atmosphere. However, their remaining nutrients requirement are obtained from the water and the mineral in the soil. Now the 11.5.1, it is the uptake of mineral ions. Unlike the water, all minerals cannot be passively absorbed by the roots. Two factors account for this. The first is the minerals are present in the soil as a charged particle, that is the ions, which cannot move across the cell membrane. And second is the concentration of the mineral in the soil is usually lower than the concentration of the mineral in the root. Therefore, most minerals must enter the root by the active absorption into the cytoplasm of the epidermal cells. This needs the energy in the form of the ATP. The active uptake of the ion is partly responsible for the water potential gradient in the root and therefore for the uptake of the water by the osmosis. Some ions also move into the epidermal cell passively. Ions are absorbed from the soil by both passive and the active transport. The specific protein in the membrane of the root hair called root hair cells actively pump the ions from the soil into the cytoplasm of the epidermal cells. Like all cells, the endodermal cells have many transport proteins embedded in their plasma membrane. They let some solute across the membrane, but not others. The transport protein of the endodermal cells are control point where a plant adjusts a quantity and the type of the solute that reach the xylem. Note that the root endodermis, because the layer of the suberin has the ability to active a transport ions in a one direction only. Now the 11.5.2 is the translocation of the mineral ion. After the ions have reached the xylem through the active or a passive uptake or a combination of the two, their functions transport of the stem to all parts of the plant is through the transpiration stream. Chief sink of the mineral elements are the growing regions of the plant and such as the apical and the lateral meristem, young leaves, then developing flowers, fruits and the seeds and the storage organs. Unloading of the minerals ions occurs at the fine vein ending through the diffusion and active uptake by these cells. The mineral ions are frequently remobilized, particularly from the older senescing parts. Older dying leaves export much of their mineral content to younger leaves. Similarly, before leaf fall in the deciduous plant, the minerals are removed to the other parts. Elements most radially mobilized are phosphorus, sulfur, nitrogen and the potassium. Some elements that are structural components like a calcium are not remobilized. An analysis of the xylem exudate shows that through the sum of the nitrogen travels as an inorganic ion. Much of it is carried in an organic form as an amino acid and related compounds. Similarly, small amount of the phosphorus and the sulfur are carried as an organic compound. In addition, the sum amount of the exchange of the material does take place between the xylem and the phloem. Hence, it is not that we can clearly make the distinction or say categorically the xylem transport only inorganic nutrients while the phloem transport only a organic material as we traditionally believe. Now the 11.6 is the phloem transport. Flow from the source to sink. The food, primarily the sucrose, it transported by the vascular tissue phloem from the source to the sink. Usually the source is understood to be that the part of the plant which synthesizes the food, that is the leaf. The sink, the part that need to store the food. But the source and the sink may be reversed depending on the season or the plant needs. The sugar stored in the root may be mobilized to become a source of the food in the early spring when the buds of the trees act as a sink. They need energy for the growth and the development of the photosynthetic apparatus. Since the source-sink relationship is a variable, the direction of the movement in the phloem can be upward or downward, that is the bidirectional, and this contrasts with that of the xylem where the movement is always unidirectional, that is the upward. Hence, unlike the one-way flow of the water in transportation, the food in the phloem sap can be transported in any required direction so long as there is a source of the sugar and the sink able to use, that is store or remove a sugar. The phloem sap is mainly the water and the sucrose, but other sugars or hormones and the amino acids are also transported or translocated through the phloem. Now the 11.6.1 is a pressure flow or a mass flow hypothesis. The accepted mechanism used for the translocation of the sugar from the sucrose to source to sink is called as a pressure flow hypothesis. As the glucose is prepared at the source by the photosynthesis, it is converted to the sucrose, a disaccharide. A sugar is then moved in the form of the sucrose into the companion cell and then into the living phloem sieve tube cell by the active transport. This process of the loading at a source produces a hypotonic condition in a phloem. Water in the adjacent xylem moves into the phloem by the osmosis. As osmotic pressure builds up, a phloem sap will move to the area of the lower pressure. At the sink, osmotic pressure must be reduced. Again, active transport is necessary to move the sucrose out of the phloem sap and into the cell, which will use the sugar, converting it into the energy, starch, or a cellulose. As the sugar are removed, the osmotic pressure decreases and water moves out of the phloem. To summarize, the movement of the sugar in a phloem begins at the source, where the sugar are loaded, that is actively transported into the sieve tube. Loading of the phloem set up a water potential gradient that facilitates a mass movement in the phloem. The phloem tissue is composed of the sieve tube cell, which forms a long column with a hole in their end wall called as a sieve plate. The cytoplasmic strand pass through the hole in the sieve plate, so forming a continuous filament. As the hydrostatic pressure in the sieve tube of the phloem increases, the pressure flow begins and sap moves through the phloem. Meanwhile, at the sink, incoming sugar are actively transported out of the phloem and removes as a complex carbohydrate. A loss of the solute produces a high water potential in the phloem. The water pushes out, returning the eventually a xylem. A simple experiment called as a girdling was used to identify the tissues up to the depth of the phloem layer. Can be carefully removed. In absence of the downward movement of the water, the portion of the bark above the ring on the stem becomes the solid after a few weeks. The simple experiment shows that the phloem is a tissue responsible for the translocation of the food, and that transport takes place in a one direction, that is towards the root. And this experiment that is performed by you easily. Now the summary of the transport in the plant. Plant obtain a variety of inorganic element, that is the ions and the salt, from their surrounding, especially from the water and the soil. The movement of these nutrients from the nutrient into the plant from the environment into the plant, as well as from the plant to their plant cell, and essentially involves the movement across the cell membrane. The transport across the cell membrane can be through the diffusion, facilitated transport, or the active transport. The water and the mineral absorbed by the roots are transported by the xylem and the organic material synthesized in the leaves in a transport to other parts of the plant through the phloem. The passive transport, that is the diffusion and the osmosis, and the active transport are the two modes of the nutrient transport across the cell membrane in a living organism. In a passive transport, the nutrient move across the membrane by diffusion without any use of the energy, as it is always done the concentration gradient, and hence entropy is driven. This diffusion of the substances depends on their size, solubility in the water or the organic solvent. The osmosis is a special type of the diffusion of the water across the selectively permeable membrane, which depends on the pressure gradient and the concentration gradient. In active transport, the energy of the form of the ATP is utilized to pump the molecule against the concentration gradient across the membrane. The water potential is the potential energy of the water molecule, which helps in the movement of the water. It is determined by a solute potential and the pressure potential. The osmotic behavior of the cell depends on the surrounding solution, and if the surrounding solution of the cell is hypertonic, to get the plasmolyzed, the absorption of the water by the seed and the dry wood take place by the special type of the diffusion is called as imbibition. In higher plants, there is a vascular system comprising a xylem and the phloem responsible for the translocation. The water, mineral and the food cannot move within a body of the plant by the diffusion alone. They are therefore transported by the mass flow system, the movement of the substances in a bulk from one point to the another as a result of the pressure differences between the 2 points. The water absorbed by the root hairs moves into the root tissue by the two distinct pathways, that is the apoplast and the sympplast. Various ions and the water from the soil can be translocated or transported up to a small height of the stem by the root pressure. The transpiration pool model is the most acceptable to explain the transport of the water. Transpiration is a loss of the water in the form of vapors from the plant part through the stomata. The temperature, light, humidity, wind speed, and the number of the stomata affects the rate of the transpiration. Excess water is also removed through the tips of the leaves of the plant by the guttation process. The phloem is responsible for the transport of the food primarily, that is the sucrose, from the source to the sink. The translocation in the phloem is a bidirectional. The source-sink relation is a variable. The translocation in the phloem is explained by the pressure flow hypothesis. Thank you so much.